Hi, everybody. Welcome to the webinar. We're going to get started in a few minutes here while we wait for everybody to trickle in. Good morning, everybody. We're going to go ahead and start and go through some of the introductory slides here and remind everybody how we run the webinar. So welcome to the wastewater surveillance webinar. Today we have two great speakers that we're excited to hear from. One is Luz de María Breton Deval from University of Mexico, and she's going to talk about her work in Mexico. And then we're going to hear from Prabhu Gounder, who is a medical epidemiologist in Los Angeles, who will be talking about wastewater and hepatitis A. Just a reminder for everybody in the audience that the Q&A is where everybody should put questions, and then there'll be a moderator for each speaker who will ask questions of the speaker. So as the webinar goes on, please put your questions in the Q&A, and you also can upvote questions there if you see one that you'd really like to ask by the moderator. A few reminders of the RCN activities. One is a journal club, which is meeting next Friday to discuss this paper by Wu et al. On measles. Tamina is going to be putting some links in the chat if you'd like to sign up and want more information. We also have another meeting of the Wikithon. So this is on Wednesday, May 14th. Again, this is a place to collect papers on WBE projects. And so again, the QR code is available, and Tamina is going to put some links in the chat. And then lastly, in June, we have our last webinar of this academic year, where Stephen Baumgartner from AAVOG is going to be speaking on looking at pharmaceuticals in wastewater and trying to understand disease burden from pharmaceuticals. And then I will also be speaking on a topic to be determined. I'm taking requests, so if anyone has anything specific they would like to hear about, you can email me and help me decide what to talk about. Okay, so with no further ado, I'm going to pass off to Luz, who can share her screen and get going as our first speaker.
Hi everyone. Just let me know if everything is in order. You can see my screen.
We can only see a green background right now.
Oh my God. Let me do it again. Now?
Yes, there you go. But yes.
Okay. Good morning, everyone. I'm Luz Breton, and before starting, I would like to thank the organizers for the invitation to present here. So I am from the Biotechnology Institute of the National Autonomous University of Mexico, and I am happy to share my experience on urban rivers indicators of environmental health and microbial functionality. Here is what I'm covering today. I'll be focusing on rivers as key sites for surveillance, highlighting their strengths and the challenges that need to be overcome for rivers to become reliable models for pathogen monitoring. As you know, water safety and quality are fundamental to human development and well-being. Ensuring access to safe water remains one of the most effective strategies to promote public health and reduce poverty especially in Latin America countries. However, in recent decades, human-driven pollution has increasingly disturbed natural environments and putting water safe at risk. Monitoring water flows is now essential, not only to detect contamination, but also to guide interventions that improve water quality. And in this context, river plays a vital role. They transport water and nutrients across continents, draining nearly 75% of the air land surface. Also, they act as natural corridors, connecting ecosystems, providing habitats for diverse species, and serve as crucial reservoir and climate regulators for human communities. Yet, despite their importance, rivers are often overlooked when we talk about safeguarding environmental and public health. In Mexico, the country has a hydrographic network spanning over 633,000 kilometers. We have particular significant 51 major rivers, which together account for 87% of the national surface runoff, of whom basins cover about 65% of the national territory. These 7 rivers are stand out because representing 71 of the Mexico total water flow. However, despite their critical ecological and socioeconomic importance, these rivers are increasingly threatened. Pollution, inadequate wastewater treatment, and uncontrolled urban discharge are putting extraordinary pressure on this ecosystem. So this led us to a critical reality. How is Mexico managing its wastewater? According to the National Water Commission, Mexico has 2,642 wastewater treatment plants with a combined installed capacity of nearly 195,000 cubic meters per second. Yet only about 57% of these facilities operate optimally. So as a result, only around 40% of wastewater is effectively treated. This means that a large volume of untreated wastewater is directly discharged into rivers. As a result, they are, open air sewage. This situation poses not only an environmental challenge, but also a significant risk to public health and microbial ecosystem stability. When rivers are turned into open sewage, the consequences ripple across ecosystem and human population alike. First, there is a dramatic reduction in biodiversity. We see loss across plants, fish, algae, a lot of organisms that support the health and resilience of aquatic ecosystems. And second, there is a concerning enrichment of pathogenic species, some of which paradoxically exhibit remarkable bioremediation capacities. This shift in microbial composition turned rivers into hotspots of microbial adaptation. And third, these altered microbial communities become powerful biomarkers indicator of both the environmental health of the river and the health of the surrounding human and animal population. Moreover, contaminated rivers become dynamic laboratories where antibiotic-resistant genes and mobile genetic elements can be studied in real time, revealing how resistant traits spread through bacterial communities. And well, the health of the river mirrors the health of the community deserves, highlighting the direct impact that environmental degradation has on public health. Well, water-based epidemiology has already proved its value as a public health surveillance tool, during the COVID-19 pandemic. It provides critical early warnings, enable the detection of emerging variants, and help guide public health responses across diverse settings. A regional analysis by Prabhu. I like this article very much. Thank you, Prabhu. And colleagues highlights the experience of Latin America, showing despite limited resource, wastewater surveillance proved be critical for monitor outbreaks and also for detecting emerging variants and informing public health interventions. However, the study also revealed an important gap. Surveillance efforts were highly fragmented and often insufficient in our region. I think if we do more surveillance, we can have more answer, and for our region are very important, this answer. If wastewater surveillance was so valuable for tracking a single viral pathogen, why we are not extending this approach to monitor a broad range of health indicators? Yes, including bacterial pathogens and antimicrobial resistance. River, after all, integrate the biological signals of entire urban and peri-urban populations, offering a unique and dynamic opportunity for early detection and public health decision-making. Leveraging rivers for genomic surveillance offer distinct advantages. First, rivers act as natural integrators of environmental signals, collecting microbial, chemical, and biological inputs from diverse urban and peri-urban areas. This integration enables a comprehensive view of pathogen dynamics and resistant patterns of the population level. Second, river-based surveillance can provide early warnings of emerging public health threats, offering a cost-effective, large-scale, and real-time complement to traditional clinical surveillance systems. Importantly, it also allows us to detect pathogens and resistant markers in communities that might otherwise be underrepresented in healthcare data. However, several challenges remain. Spatial and temporal variability in river systems can complicate interpretation of microbial and genomic signals. For that reason, we need to standardize sampling protocols across diverse hydrological and population contexts is critical to ensure comparability and reproducibility. Now, given this background, our study aims to explore several questions. Rather than list them all, let me highlight the following question. Can the analysis of wastewater in rivers serve as an effective model for monitor population health? Now, let me share with you some of the main findings from our study, highlighting how population shapes microbial communities, functional potential, and resistant dynamics in urban river systems. Our first case involves our findings in the Apatlaco River, one of the most important water bodies in our state, because it provides water to nearly 2 million people, many of whom live along its bank and are directly exposed to its current conditions. Unfortunately, the river is severely impacted by more than 320 discharges and urbanization. To better understand the environmental and microbial consequences of this pollution, we analyzed 17 sampling points along the river. We measure a range of physicochemical parameters, including coliforms, chemical oxygen demand, nitrogen species, phosphorus, and dissolved oxygen. Yes, I show here some of them, of these measures. The highest population levels were recorded at sample point 10. This sampling point was the highest for cold, with 480 milligrams of cold. And we also found 70 milligrams per liter of nitrite and 2 milligrams of dissolved oxygen. This is very low level of oxygen, and nearly 300 milligrams of total dissolved solids. So these values indicate extreme environmental stresses. And to explore microbial dynamics, we select 4 representative sites and perform metagenomic shotgun sequencing. Here, this is our selected site. Here, we want to go deeper and understand the microbiome. So, Protobacteria was the dominant phylum. However, we observe a clean contrast in microbial composition between or across all the sites. The clean water reference was enriched in freshwater planktonic bacteria like Linovitans, while polluted sites from here to here were dominated by potential opportunistic pathogens such as Acinetobacter, Arcobacter, and Myroides. These findings highlight not only a loss in ecological quality, but also a serious public health concern for the million of people who rely on this river. And this case demonstrates how environmental degradation directly affects microbial composition and increases the risk of waterborne disease. As a continuation of our work of the Patlaco River, we investigate how specific discharge influence microbial dynamics and resistance profiles. Rivers face intense anthropogenic pressures from multiple water inputs: agriculture runoff, hospital effluents, domestic wastewater, and treatment discharge, all of which are alter the microbial community structure and potentially enhance the spread of antibiotic-resistant genes. To characterize these impacts, we analyze samples from different types of discharge. Here I put an image. And each type shows a distinct microbial profile and also a resistant signature. It's very interesting for us that hospital wastewaters, for example, harbor microbial genera. This is microbial from the hospitals, associate with treatment plants because the treatment plant of the hospital was anaerobic, so we found a lot of these kind of microorganisms. But the most interesting thing is that these samples also, we were able to isolate a multi-drug resistant strain of Klebsiella aerogenes. Here I show the Klebsiella that we can isolate. The most interesting thing is this Klebsiella was a multi-resistant bacteria. What else? Well, in untreated domestic wastewater, that was this, we detect a dominance of Arcobacter, Acinetobacter johnsoni, and Pseudomonas sutseri, indicating incomplete removal of potentially harmful bacteria. We also detect Acinetobacter uofi. When it comes to antibiotic-resistant genes, hospitals' effluents contribute significantly to RG diversity, including genes conferring resistance to last resort antibiotics like meropenem and vancomycin. We also can observe that in this last point of the river, the same kind of bacteria, for example, Escherichia coli or Arcobacter, but also some freshwater microorganisms like limnohabitants. But these pathogen bacteria that were very, we found them with high levels in the previous discharge. We also found them in the last point, and these results from the environmental led us to consider rivers as potential receptors of biomarkers that could provide insights into population health in a cost-effective way. While clinical epidemiology is highly accurate, it is also very expensive. Although such studies are conducted in Mexico, they are relatively rare in a country with such diverse realities, you know? That is why we have decided to continue working in rivers with a focus on public health. While the Apatlaco River helped us to explore the use of rivers as low-cost health monitor system, the environmental and social complexity of the area poses several challenges. Therefore, for our second case study, we chose a setting with more control. And this site was the Magdalena River, a unique location within Mexico's hydrological and public health landscape. It is the last remaining urban river in Mexico City, a metropolis of over 20 million people. What makes this site particularly valuable is that the sources of the sewage are well identified. And well, Mexico City publishes epidemiological reports that are publicly available, allowing us to correlate our findings. So through a full year, we conduct a longitudinal metagenomic survey of the river wastewater microbiome, focusing on pathogenic bacteria with high national and international epidemiological relevance. Here I show a image where I show the selection. This selection was based on three established frameworks: the WHO Priority Pathogen List, the escape group, and Mexico National Surveillance System. We identified clinically significant species, including Staphylococcus aureus, Vibrio cholera, Neisseria meningitis, Clostridium tetani, Klebsiella pneumoniae, and Escherichia coli, all showing increased abundance in summer. Here I put these results. We also observed seasonal shifts in species like Enterobacter. Here is Enterobacter, Streptococcus, and Acinetobacter during spring. Acinetobacter johnsoni rose from 7% in spring and summer to 60 let me found Acinetobacter johnsoni. Here is here is another Acinetobacter. We also, for us, was very interesting, found Aliarcobacter creaerophilus. Aliarcobacter, here it is. Because it's a foodborne pathogen, these kind of microorganisms indicate to us that there is something, like a chicken farm a clandestine chicken farm. So this kind of analysis also or let us to identify this kind of extra, extra information or this yes, this relevant uh, illegal discharge. So what else? Well, our findings, to complete these taxonomic findings, we performed resistant profiling using a sample called contents and detect resistant genes against 24 antibiotic families, 20 of two which were consistently present throughout the year, and the highest number of resistant associated genes appear in summer, followed by winter. These include genes linked to resistance against aminoglycosides, cephalosporins, fluoroquinolones, macrolides, penicillins, tetra tetracyclines, phenycols, and linctosamines. Our
Hi,
findings.
sorry, we have some questions and I want to make sure we have some time to go into questions.
Oh, sorry. Yes. Okay.
Yeah. Um, thanks. Okay. Well, I have a question actually that was on your you had a kind of advantages or challenges or things to consider with respect to river monitoring, and one of the things was thinking about how you normalize these sequencing signals. And so I was curious when you're thinking about seasonality, how you're thinking about understanding what these relative abundances mean across these different seasons.
So the question was that how I normalize this variation between the seasons.
Yeah, and all the other inputs. We also had a question about like decay of the signal over time as well.
Okay. Well, rivers are very complex because we only measure superficial water, but we also need to consider in the future the sediments. Um, we also will have some microorganisms that will be there in the sediment and could be a constant point of yes of liberation of microorganisms, but we have some controls, and also we use this kind of communities, mock communities, to establish the protocols and to calibrate the equipment. This kind of controls can help us to have a better signal of the different microorganisms that we can find between the different seasons. So for us was very interesting that some of these microorganisms are are consistent or are yes consistent with with some reports of epidemiological with the epidemiological reports of our city. For example, if we found more more abundance of Enterobacter pyogenes, we also found more abundance of intestinal illness. However, in Mexico, we have some problems with our epidemiological surveillance because most of them are very general. Like for example, most of the cases are enteric and respiratory, and our our idea is that our system is more accurate because it's not only enteric illness in general. You can detect more cases of these are people that doesn't go to the hospital. So
Yeah.
the tracking is more, like more accurate.
Awesome.
Yeah.
Well, this was a really good presentation. There are some questions in the Q&A, but I think we probably need to go to the next speaker.
Oh, sorry.
Oh, it's totally fine. If you wouldn't mind answering maybe those, and we'll have Rolf take it away or go to Prabhu. Thank you so much.
Thank you.
Thank you, Hesed. Great talk, Luz. I think it's a really interesting application. Yeah, hopefully we can have some exchange here by email to answer those lingering questions. Next up, we have Prabhu Gounder, who is going to talk to us about genomic and wastewater testing data to inform a hepatitis outbreak in LA County. And he joins us from the LA County Department of Public Health. Prabhu, take it away.
All right, good morning, everyone, or good afternoon, where you may be. Am I showing the right version? I want to make sure it's the slides and not the presenter view. Okay, great.
Look good. Single slide to be seen.
All right, great. Well, today I wanted to share our experience using genomic and wastewater testing data to inform an ongoing hepatitis A outbreak response in LA County, and this outbreak started in 2024. Here. Great, so just a quick background for people who are not familiar with hepatitis A virus as an infection. It's a liver infection that's caused by the hepatitis A virus. It's transmitted by what we refer to as the fecal oral route, which means it involves ingestion of food or water with microscopic contamination of feces from an infected person. And this commonly happens in areas without adequate access to hand sanitation or disinfection. So they don't wash their hands, eat food after, and they can get infection. It can also be associated with close personal contact, such as sexual contact or sharing of drug use equipment. It is incredibly transmissible, and there are a couple of reasons for it. First is that it can persist in the environment for a very long time. It's really hard to eradicate. It can persist for up to months in the right conditions, and you need stronger agents like chlorine bleach to get rid of it. So it's possible for people to become inadvertently exposed to this, weeks after a surface is contaminated. And it also has a very low infectious dose, just, very few virions to set up an infection. So I think that combination makes this a very easily transmitted infection, but it is highly preventable. There's a very safe and effective vaccine. And the same vaccine, if it can be given within 14 days of last exposure, it can also decrease the risk of getting infection. So in LA County, we conduct surveillance for about 10 million residents across 86 cities. And hepatitis A is reportable by local mandates by clinical providers and clinical laboratories. We started receiving wastewater testing data for hepatitis A in September of 2023, and this is testing done by Verily. They are testing at two of the largest treatment plants, one at Hyperion in the city of LA, and the other at Warren, which is with the LA County sanitation districts. The map on the right shows sort of a larger peach-pink area, which is LA City, and then a little bit below it to the right is that bluish gray, which is the Warren distribution. So there are some challenges with clinical surveillance for hepatitis A that's important to understand for context in interpreting the wastewater data. The first is that 30% of adults are asymptomatic, and meaning they will not present for care, so there's no opportunity for us to detect them by surveillance. But even when people do present for care, the symptoms of early infection can be nonspecific, include things like fever, nausea, vomiting, abdominal pain, and diarrhea. So it really just looks like the stomach flu or gastroenteritis. So a provider might not think to test for hepatitis A. But even when they do, the test that is required for diagnosis is the anti-hepatitis A virus, IgM antibody test. It false positive in up to 90% of symptomatic cases. So it requires a little bit of judgment, looking at the medical records to make our best assessment of whether we think this is hepatitis A. But this does lead to a certain amount of misclassification of cases as well. And then finally, there's some challenges with both the case investigation and management of contacts. So the incubation period is that time between when someone is exposed to the virus and when they develop symptoms. And for hepatitis A, it's up to 7 weeks. And when you have such a long incubation period, it becomes really challenging to figure out when, where, and how someone acquired the infection, especially when they're unhoused and you don't exactly know where they were and what the nature of the contacts are. The infectious period, so that's the time when someone is shedding and could spread the infection to other people, that it starts 2 weeks before symptom onset and can last up to 1 week after. So that means a lot of the transmission actually occurs before we can even intervene on the public health side. So now to turn to our outbreak, we had 3 cases reported in persons experiencing homelessness or PEH back in April of 2024. And when we got these cases, it wasn't clear, these numbers are small, does this represent an outbreak or is this just a coincidence? So then we looked at the baseline. So going back to September of 2023, when we started getting wastewater testing data, we see that there's typically about 2 to 5 cases per month, and in March and April it was about the same. But what was notable is that this was the first time in several months that we've had cases within persons experiencing homelessness. So again, not clearly above baseline, but concerning for a potential cluster. So then we turned to the wastewater data, and again, we didn't really have much of a baseline before September, so we didn't know what to make of these concentrations. So we were mainly observing it until April, when we saw a sharp increase in the wastewater concentrations. I forgot to mention the yellow color represents LA City, Hyperion. The purple line represents Warren. And all 3 cases were within the city of LA's wastewater distribution. So when we saw this sharp increase in wastewater concentrations in conjunction with these 3 cases in a very defined population in a defined space, this made us concerned for broader undetected transmission, which makes sense for people who are unhoused and may not have the same access to healthcare. Also, we in LA County try to routinely obtain specimen for sequencing, and all of 3 of the PH cases had the same novel strain. And this is a strain that was reported for the first time in public databases from LA County. So this further supported our concern that they're part of a common chain of transmission. So this led to our decision to activate a response. And at a high level, what the response looks like is enhanced case findings. So instead of passively waiting for cases to be reported, we actively tried to identify them, conducting outreach, vaccine outreach to PH service providers, conducting environmental health assessments at shelters, encampments, and restaurants, extensive communications with providers and community partners about hepatitis A so they can maintain vigilance and recommend vaccination. And historically, we've closed the outbreak when there's no new cases reported for 2 incubation periods. So that's up to 14 weeks. And the reason for that is because of the asymptomatic cases and lack of diagnosis. There's a possibility that you could miss a case if you just wait 1 incubation period. So moving forward, in the next 5 months, we identified an additional 33 cases. And these 33 cases, 21 were among people experiencing homelessness or using illicit drugs. There was a lot of overlap between these 2 risk factors. And we also identified 12 cases that were not among people with risk factors, and we were able to link them by sequencing. So they had that matching novel strain, which again helped us tie them to the local outbreak as locally acquired cases. We also continue to see the wastewater concentrations fluctuate, but it never reached the peak that we saw in April when we started the outbreak. And we see some of the challenges here with interpretation. At the end of April, beginning of May, the wastewater concentrations were similar to what we're observing in September, but you can see there's many more cases reported in September compared with May. And we don't know, is it because of our enhanced case finding efforts were more successful, so we're able to link more of the cases? Is it because the cases in May were just shedding more per case than the cases in September? So, it's hard to assess that. And the other challenge here too, at least when we're looking at this initially, is we did not, and this is not coded by sewer systems. So it's possible that some of these cases occurred outside of the sewer system, so wouldn't be contributing to the wastewater data. So as a next step, we wanted to try to take a deeper look to see if we try to correlate the occurrence of hepatitis A detections in wastewater with clinical surveillance. And so this is a figure where each bar represents a date that a wastewater sample was collected. The sort of green teal color represents dates when there was a hepatitis A detection. The yellowish tan lines represent dates when there was no hepatitis A virus detection in the wastewater. And then the red lines, the horizontal lines, represent the cases. So this is specific to LACD Hyperion. We could do the same for Warren, but just wanted to share this as an example of how we're thinking about the next steps and further evaluating this correlation. The line extends for 3 weeks because that's the hypothesized period of shedding and infectiousness. And the red circle represents the date when we think someone developed symptoms. So that is the 2 weeks after or 1 week before the infectious period. So, you know, one of the things that we note here is there are times where you can have a clinical case without, you know, corresponding wastewater detections. And I show that with the red arrow there. And some of the potential reasons for this could be that the hepatitis was caused by something else other than hepatitis A. And as I mentioned, 90% of the IgMs are false positive, and people frequently have 2 different causes of hepatitis. So it's possible that you know, we call this you know, a hepatitis A when it's really due to something else. The other possibility is that it's delayed. There's a delayed diagnosis. You know, we have identified some cases who were tested as a result of our outbreak declaration, but weeks after they had the infection. So it's possible that we just placed that case on the wrong part of this timeline. As I mentioned, some of the cases in the previous figure could have resided outside the sewer system, but even when we geocoded, you know, the way we assign cases is based on residential address. So we had at least a couple of cases where they were exposed in the sewer system, but they were traveling outside of LA County when they became symptomatic, but they would still be assigned as a case to LA County and they wouldn't contribute to the wastewater detection. And then the last possibility is just that the number of cases is below the level of detection. We actually don't know how many cases you need to have a detection in these sewer systems with, you know, 4 million people represented. Conversely, we also have situations where there's wastewater detection without a corresponding clinical case. And then some of the reasons for this, and again, I've highlighted that with the red box here. So you see a couple of areas where there's the teal detections without any overlapping cases. And so one reason is that there is an undetected case, that the person was either asymptomatic or the provider didn't think to test. This could also represent misclassification in the opposite direction. One of our criteria in the case definition is that the IgM is positive in the absence of an alternative diagnosis. So oftentimes when there is an alternative diagnosis, we typically would consider this to be not a case. So that could be a situation in one of these 2 areas that I've highlighted. And then the last situation is that the case is a resident of a neighboring jurisdiction. So if someone is on the border and they have a residential address on the border, but they work or spend substantial time in LA County, they would contribute to the wastewater, but you know, they would not be reported to us and we would not know that they were a case. So in terms of the next steps, you know, we wanted to review the medical records of the reported and confirmed cases to better understand the reasons for discordance, to really explore those hypotheses that I stated in the previous slides. We will soon have the ability to do sub-sewer system level testing. So this can help us further evaluate whether some of this discordance is due to a limited detection. We can potentially, you know, if we do find cases in a smaller sewer system but not the larger sewer system, we may be able to evolve to an understanding of the number of cases needed for detection based on the population represented. And I think we do need to try to understand sort of the duration of shedding to better correlate in how long we'd expect to see a detection in wastewater. We also think whole genome sequencing wastewater can help infer the proportion of hepatitis A cases that are attributable to an outbreak. We historically prioritized outbreak-associated cases for sequencing. And so when we started to see all of the cases associated with this new strain, it wasn't clear to us whether it's because we were selecting for those cases or whether this was the prevalent strain. And so if we're able to distill that in the wastewater by sequencing, that's something that would help us better understand our surveillance data. I think there's still going to be continued challenges with wastewater and clinical case correlation. We're not always going to be able to exclude alternative causes. There's often multiple causes present. Could be cirrhosis. People with severe hepatitis A infection can present like they have sepsis. We've had a couple of cases of autoimmune hepatitis that were triggered by hepatitis A virus infection. And so without PCR testing, you're not necessarily going to know what the cause is. We also will continue to have some incomplete information on the body site, duration and magnitude and variability of shedding that can infer our ability to correlate concentrations with the number of cases. I think some of the potential solutions for this would be modeling. So if we have some additional information, potentially model to account for asymptomatic cases, if we assume that it's non-differential over time. And PCR is something that is not routinely done. It's not a clinically available test. We're lucky to live in LA County, where our state is able to offer this service of PCR confirmation plus sequencing, but it's not something that is routinely available. So that does make it hard to mitigate misclassification. But a preliminary conclusion so far, we can see that increases in wastewater concentrations are likely to represent undetected transmission. I think it's helpful, especially early in an outbreak, to know if there's increased community transmission. The decreases in wastewater concentrations are less reliable because you can miss transmission that occurred within the sewer system boundary, but if the case resides outside of that boundary. But still, having no detection for an extended period of time could support our ability to close outbreaks earlier. Maybe we don't need to wait 2 incubation periods. Maybe we can close it in 7 weeks if we don't see a wastewater detection suggesting undetected transmission. But until there's this improved understanding of that correlation between changes in wastewater and concentrations and the case incidence, I think wastewater surveillance is really just going to be a tool that's best used to complement our traditional case-based surveillance strategies. So I'll stop there, and I'm happy to take any questions.
Thanks, Prabhu. Wow, that was very compelling and interesting, and also challenging, I think. Looks like this is a very difficult application for using wastewater-based epidemiology, but it's an important topic, obviously. Let's see, we have one question was related here to where the sequencing data was obtained. Can you tell us? I think you mentioned the LA County lab was doing the sequencing, and then also the method that was employed.
Yeah, so the sequencing is done at the state, and they use 2 different methods. The quick version that they've been doing routinely is Sanger sequencing of a specific hypervariable region of the genome. But more recently, they've been doing whole genome sequencing. And now we have about 70 cases that have been sequenced, and it's been really informative for us to look at that phylogenetic tree. So I know this is a webinar on sequencing, so on wastewater testing. So I won't go too much into what the sequencing found, but that's the approach at the state.
Okay. Another question is, what are your thoughts on how to validate wastewater surveillance for diseases where clinical case data are poor, like for hepatitis A? Is it possible?
I think so. I don't think we're going to be able to get to the sort of correlations we're able to do, say, with SARS-CoV-2 at a time when the only testing was PCR, so all of it was reported, and then we had wastewater concentration, so you're able to do more precise correlations. I'm not sure we'll ever be able to get to that point, but I do think we can do better than where we are now, which is a complete absence of information. We're just left with the very qualitative correlation between wastewater, and qualitative, I mean increases mean there's a detection, decreases mean that potential transmission is going down, but no ability to understand that in a more quantitative manner. I think the next step for us would be to do more systematic chart reviews. We have since the outbreak started, about 700 reported cases, of which we've confirmed over 160. And so I think what I'd like to do is, in that figure that I showed you, those time periods when we don't have any reported cases, but there's a wastewater detection, we can go back and look at the reported cases as well and try to pull those records and see, did we potentially misclassify the case? Conversely, the issues that I talked about in terms of a case that was asymptomatic and detected, but because we didn't know exactly what the symptom onset date was, we used the lab test date, so that could throw off that correlation. But again, we can go to the records and we know things about the patterns of what the liver function tests look like. There's a increase in the ALT, decline, a delayed increase in the bilirubin. So if we can look at the records and see what that pattern looks like, we could try to maybe work back and say, actually, based on what we know about these patterns, the likely onset date was maybe 2 to 4 weeks earlier. The one benefit that we have is we also have PCR confirmation for a lot of these cases as well. So that does help us mitigate some of the misclassification. So when we did misclassify a case, we're able to then get the PCR results and reclassify. So we can go back and look at those cases and see what we can learn. So I think we are working on kind of more streamlined abstraction form to look at risk factors, the lab tests, that type of clinical information to help us refine a pretest probability of a case and see if we can get more precise information about the occurrence of cases in relation to wastewater testing.
Yeah. And it looks like if you don't have a signal, it doesn't mean that you have absence. Absence of evidence is not evidence of absence. But if you do see a signal in the wastewater, that obviously motivates one to look for those cases that have not been reported in the health system. One question is, I appreciate LA. It is not just a big city. It has a lot of wildlife, and it's a really great place. And if you go out camping just far out of the city limits, you might encounter mountain lions and snakes and all kinds of things. So are there animals and mammals that carry the hepatitis virus or are genetically related to pollute the signal?
That's a good question. Now, I don't know. Actually, I haven't looked at non-human source of hepatitis A, but I think there are. I've heard of hepatitis A outbreaks from berries that are contaminated by wildlife being improperly washed.
Oh,
so I do
okay.
think that does exist, but in LA, there's a closed sewer system, so we don't get animal sources. It's really just plumbed water. Occasionally there may be heavy rain and some flow that gets diverted to some of the surface runoff, but for the most part, what we're seeing is human contributions only.
Yeah. And I think for the COVID applications, we often use the COVID signal in wastewater to then zero in on areas and find cases, right, and focus our interventions there. It seems to me that all the detections that you did were cases that were medically, clinically reported, right?
Yeah.
And so there is no immediate kind of geographic information really flowing other than the number of cases. Is that fair, or were you able to pinpoint locations where you then intensified your screening?
That is something we want to do. Our limitation was that this is testing that wasn't done at the direction or request of LA County. This is something that was done, by Verily to support improved wastewater testing. So certainly we're grateful to have that information, but we also didn't have the ability to independently test at sub-sewer systems. But we did receive funding in the last year. So we're now going to be a locally funded jurisdiction by CDC for wastewater testing. And we're working towards, and we have started actually collecting wastewater data at sub-treatment plants. Our goal is to do that testing in those sub-treatment plants to try to better understand some of these correlations.
Okay.
And yes, and moving forward, you're right. And once we have that in the setting of an outbreak, if we have a detection in a specific area without cases, it would potentially allow us to do some additional messaging to increase awareness among providers to consider testing.
Okay, so I have one quick question, and then I might want to take the last 5 minutes to talk about the vacation period that's coming up and how the information you dispensed might inform our behavior as travelers. But here's a question regarding, so we are intrigued by the study, and we're wondering whether other viruses other than hepatitis are kind of on your list, or you think are low-hanging fruit that could be monitored by wastewater monitoring, but haven't been explored yet?
Our goal is, and I think we might have colleagues from Zymo on this call, but we're partnering with them to really do wastewater testing for every reportable infectious disease and every pathogen for which we have clinical data. So we're looking at over 100 different targets. I think one of the things that I've realized from SARS-CoV-2 and also with hepatitis A is that, there's a lot of work. Of course, there's a lot of work on the wastewater side in terms of collection standardization and methods, but for it to be useful for public health, there's still a lot of additional work that needs to be done. So using SARS-CoV-2 as an example, people look at the wastewater data and our rolling averages, and it's pretty intuitive, it matches well, and they think it's easy, but it wasn't, right? We had to think about for the wastewater concentrations, we tried different approaches, weekly averages, 10-day averages. We decided to eventually land on one where we dropped the highest and lowest within a 10-day period and take a 10-day rolling average, and that seemed to work well. There's a lot of trial and error that went into figuring out what's the right smoothing approach to account for the variability and account for those outliers. And then to do that work, and that was done by the wastewater scan group about to show that it actually does correlate with cases. With hepatitis A, we're starting to see some of those challenges now. We still don't know the pathogen now that we think sheds for up to 3 weeks. What is the right smoothing approach? And there's even more variability for hepatitis A than there is for SARS-CoV-2. So we're going to have to go through that same process, and to do that requires some of this additional chart review that I talked about. We're also looking at measles. There's different cases. There haven't been detections until recently. Why is that? We know that people are getting vaccinated all the time. It's a live attenuated virus. Why aren't we picking it up? We're looking at dengue separately, and I know there's a separate webinar by RCN a couple of weeks ago where we have DNV3, which is very common, but we're not picking it up in the wastewater, but we're picking up uncommon DNV1, 2, and 4. So those are just examples where I think each pathogen is different and requires individual in-depth evaluation before it can be useful.
Thanks. We are running out of time, but I have one more question for you for those who go on vacation. You kind of scared us. You said this is a virus that persists for long periods of time, short of chlorine, you can't, knock it out. And, the onset of disease is late. It's a serious infection. So the vaccine you mentioned, how long does that last? How long does that protect? And is it easily available? How is it administered?
Yeah, so I think part of the reason we're not seeing a lot of outbreaks, despite it being highly infectious, is there's two parts to this. Older adults are mostly immune, similar to like childhood immunization, childhood illnesses. A lot of people are exposed to these epidemics, in their childhood, and they're immune. There's another group of people that have been vaccinated. So the vaccine was licensed in the early 1990s, and it became part of the routine childhood schedule in the US, at least as of 2005. So every child now is getting vaccinated. So there's a large amount of population immunity already. The vaccine is 2 doses, 6 months apart, highly effective, greater than 95, 98% effectiveness, lifelong immunity. In the context of an outbreak, even a single dose can lead to greater than 90% immunity at 4 weeks. And protection is thought to last up to 10 years with just a single dose. So you really need that second dose for lifelong
So I think
immunity.
a lot of us who listen to your talk feel a little better now. We have 1 minute left. Maybe Tamina can flash those slides again that we had in the beginning. I just want to point out future activities. So on May 9, just a week from today, we have at 3 p.m. Eastern Daylight Saving Time, the Journal Club coming up. And then on May 14, we have the Wikithon. Hope you guys can join in and help us populate our Wikipedia. So that's again at 3 p.m. Eastern Daylight Saving Time. And then our very last webinar is on June 6, featuring a speaker from Airwalk and then our very own Alexandria Bohm. So thank you, Prabhu, and thank you, Luz, for joining us here and presenting. It's been great. Tamina, Nick agrees. And Tamina, do you have any further announcements, or should we let the audience go?
Yeah, I think we're done. Thank you.
I think we're good.
All right. Thanks for great talks. Appreciate it.
Thank you.
Thank you.
Bye.